As we look around us in this complex world, we see information everywhere. From the cars we drive, to the mobile phones we use, to the image you're watching right now. Information is everywhere and is an essential part of our lives. We're drowning in information and yet starving for knowledge. With so much information all around us, have you ever stopped to ponder the information of life? The code that's in every cell, every piece of DNA, and all living things. Hi, my name's Chris, and we're going to dive deep into the world of cellular biology and information science to see if we might get a little closer to the big question, where did we come from? You know, even though we've made many breakthrough discoveries in science, there's still so much more to be discovered. It will be through these discoveries that we'll be able to cure disease, improve our well-being, and possibly discover the origin of life. And with the most common worldview of life's origin being chemical evolution, how does this scenario align itself with our knowledge of information science? Information science is changing the way that we think about all living systems. So what is information? Is order and organization information? When we look at nature, we see a lot of organization. Salt crystals, stalactites, even a sand dune could be considered organized. However, these would be considered examples of self-order. What about a simple snowflake? Is there order to its structure? Look under a microscope and see in amazing detail these exquisite patterns. Water, cold air, gravity, and time gives you snowflakes. In a cavern, water, minerals, gravity, and time equals stalactites and stalagmites. How about a tornado or a hurricane? These are powerful self-ordered forces, but do they contain meaningful information? The answer is no. They do contain data, which is determined by physical constraints, but not information. The behavior of these things are governed by what we now know as the chaos theory, which simply stated is the study of how order forms naturally without design. The question is, can information form by chance? It seems that things containing information are highly organized, but there's more than just order. Much more.
Information is intangible because there's always a sender of the information and there's always a receiver of the information that has some ability to decipher the information, decipher the meaning behind the information that's being sent, and then changing into some meaningful, useful purpose.
Information must have a sender and receiver in order to communicate. For instance, these flashing lights have no meaning to us. But what happens when we place them in a straight line and add color? Now we recognize the code that's being transmitted, directing us to stop at red and go on green. This preconceived knowledge is important, and it makes a big difference in our everyday lives. In fact, without this knowledge, our travels would become quite chaotic and dangerous. So you see that even a simple code like these traffic signals can be considered information if knowledge is sent and received. In a moment, we'll discuss the information in living systems, but first, let's discuss three main categories of information that scientists use. The first is called Shannon information. Shannon information provides only a mathematical measure of probability, like if we rolled a die 100 times and recorded the results. This would give us data, but it would be random data, not useful information. The second category is called functional information. This is information that's useful and practical, like our traffic lights. Information is sent by the source and understood by the receiver in order to ensure safe travel. The last category is called prescriptive information. Prescriptive information is what we're going to be primarily focusing on. It's instructional information used to determine what choices to make and could even be based on a record of choices already made. In order for something to be considered prescription, the receiver of the message must have the knowledge of the source's alphabet, rules, and encryption in order to decipher the message and act on it. For instance, have you ever seen what software code looks like? At first glance, it just looks like a bunch of random characters. However, If you know what those characters represent, and you have a system to decipher the code, you can understand and create extremely powerful prescriptive programs. Think about it. What at first appears to be just meaningless ones and zeros is actually information, knowledge, just waiting to be interpreted and executed. It's incredible to think about how much information exists all around us. New York City is a pretty busy place. The traffic, the people, the messaging, the systems of transportation. But when comparing a living cell to this great city, New York pales in comparison. A cell is so complex that scientists have only scratched the surface of what really goes on in there. However, even with our limited understanding, what we do know is that the inner workings of a simple cell are simply extraordinary.
It's almost like there's a miniature city, and every cell, and everybody has their own little job. If all the traffic lights decided to all just go crazy, you have issues that not only do we fix the light, but also we have traffic issues, we have crime issues, we have problems that snowball from one part breaking down.
The cell is an extremely efficient and well-networked computer system with thousands or even millions of processors and billions of bits of information. Cells are the structural and functional units of all living organisms. Some organisms, such as bacteria, are unicellular, consisting of a single cell. Other organisms, like humans, are multicellular, having many cells. In fact, it's estimated that an adult human has over 100 trillion cells, and each cell is an amazing world of its own. It can take in nutrients, convert the nutrients into energy, carry out specific functions, and reproduce itself as necessary. Even more amazing is that each cell stores its own set of instructions for carrying out each of these activities. Life requires fully functional DNA, RNA, ATP, enzymes, and other proteins. If any of those components were missing or not working properly, life would fail to exist.
Hello, I am Dr. David Bick, and I'm going to continue my discussion of whole genome sequencing. This is one of a series of short videos on this subject. In this segment, I will discuss genes, DNA, and mutations. In a previous segment, I discussed chromosomes. But what are chromosomes? They are the structure that contain our genes. Think of chromosomes as strings. Genes are like beads on a string. A chromosome has a few hundred to a few thousand genes on it. In total, we have about 20 to 25,000 genes. What are genes? Genes are instructions that determine all of our features, such as eye color, hair color, skin color, height, and so forth. Again, our genes are part of the chromosomes, and we pass them in the egg and sperm. This is how we inherit our genes, and this is why we resemble our parents. Genes work. I like to think of genes as recipes in a cookbook. They tell our cells how to make things. For example, genes tell our cells how to make proteins. It is the proteins that do the work of the cell, such as breaking down our food to supply our body with energy. When our cells need to make a protein, it reads the information in the gene like reading a recipe in a cookbook. If there are mistakes in the gene, then the body can't make the protein, or it makes it incorrectly. We call these gene mistakes mutations. To make this clearer, let's think of a recipe for brownies. If the recipe calls for 1 cup of sugar and you add 8, the brownies will not come out correctly. It is the same with genes. Even one tiny change in a gene will result in a problem. But what are genes made of? Genes are made of deoxyribonucleic acid, or DNA for short. And DNA is made of nucleotides. There are only 4 kinds of nucleotides: A, C, T, and G. Think of them as 4 letters of the DNA alphabet. A string of these letters will make a gene, and when the genes are strung together, they make a chromosome. All genes are composed of just these 4 letters. The order of the letters and the number of letters is different for each of our 20 to 25,000 genes. To follow the recipe idea a little further, if genes are recipes, then nucleotides make up the letters and words of the recipe. So a mutation in one nucleotide of a gene or one letter of a recipe can result in an abnormal protein or, as mentioned before, ruin a batch of brownies. Remove the middle section of a gene or the middle page of a recipe and the results are worse. There will be no protein, or in the case of the recipe, no brownies. Thank you.
This animation demonstrates how the digital information encoded within DNA is used to direct protein synthesis. This is a DNA double helix containing the digital code which directs the cell in all aspects of operation. And here we see a protein complex called an RNA polymerase traveling down the DNA strand. As it moves down the strand, it carefully unwinds the DNA, preparing it for transcription. Inside the polymerase, we see a single-stranded copy of the original instructions being assembled as individual bases are positioned and added to the growing strand. A stop code marks the end of the protein specification, at which point this copy, known as a messenger RNA transcript, exits the polymerase and heads towards a two-part chemical manufacturing machine called the ribosome. While the messenger RNA moves towards the ribosome, transfer RNA molecules attach to specific amino acids in preparation for assembly. As the messenger RNA transcript passes through the ribosome, the process of translation begins. Using the instructions encoded on the messenger RNA as a template, the transfer RNA molecules align specific sequences of bases to corresponding amino acids, creating a protein chain. As this chain exits the ribosome, it is met by chaperones which prevent premature folding, while escorting the protein to a barrel-shaped machine called a chaperonin. This machine helps fold the protein into the precise shape required to perform its function. Although it is unclear how the chaperonin achieves this, we do know that accurate folding is essential in order for the protein to accomplish its intended function. Once the protein is complete, it is released into the cytoplasm to do its job.
As you can see, protein synthesis is an amazing process which occurs constantly in our bodies. Now, get this. Ribosomes are made from RNA and proteins, so the ribosomes are made of the very proteins they manufacture. In fact, it takes over 150 existing proteins to manufacture just one protein. So let's examine what DNA is and how it works to better understand how information is used in the cell. Deoxyribonucleic acid, or DNA, is a long polymer of nucleotides, each nucleotide being a deoxyribose sugar molecule, one phosphate group, and one base. Nucleotides are joined together to form the DNA backbone. A second DNA strand then joins the first to form a double helix. You may recognize the code letters A, C, G, and T, which are the four bases used in DNA. These bases are adenine, A, cytosine, C, guanine, G, and thymine, T. Like the code in Mr. Gates' software, the sequence of the four bases encodes genetic instructions used in the development and functioning of all known living organisms. Once organized, this information is what makes up the gene. And genes, as we all know, contain the recipes for the components of life. Genes hold all the information to build and maintain an organism's cells, and they also pass genetic traits to offspring. Human DNA contains an estimated 20 to 25,000 genes in its approximately 3 billion base pairs. And did you know that if the DNA in an adult human's body were laid out end to end, it would be about 30 billion miles long? That's enough to travel the distance from the Earth to the Sun over 320 times. A gene contains both coding sequences that determine what the gene does and non-coding sequences that determine when the gene is active, as well as thousands of other regulatory functions. Less than 2% of the genome is for coding proteins. The other 98%, which used to be thought of as junk DNA, is now being studied to determine its functionality.
Chemically, the DNA in the cells of my body is the same as that in a fruit fly, an orange, a hippo, an elephant. It's all the same chemically. The uniqueness to all of those species of living things lays in the information content of the DNA. So while the chemistry may be the same, the actual information that's embedded in that chemistry is very, very different. And the challenge for molecular biologists such as myself is understanding completely what that information means.
When a gene is active, the coding sequence is copied in a process called transcription, producing a messenger RNA or mRNA copy of the gene's information. mRNA is produced by a protein that's over 3,000 amino acids long as it reads the genetic code. Wait a minute. How can messenger RNA require a protein that reads the genetic code, but yet the protein needs the messenger RNA to be produced? Could these have both come into existence at the exact same time with the meaningful information needed for both to operate properly? As you can see, much is still a mystery when it comes to the origin of this complex organic language.
And what's fascinating to me, not only do proteins create proteins, but we have proteins that are actually creating the DNA molecule. That's our blueprint. And yet on the DNA molecule, there are sequences of part of the DNA that are coded for those proteins. So the manufacturing plant needs the blueprint, and the blueprint, the DNA, needs the manufacturing plant in order to actually create the DNA. So once
DNA is organized into a single coiled piece, it's then wrapped around a complex hierarchical structure of proteins. This is what is commonly known as a chromosome. There are 23 pairs of chromosomes in the human genome. A genome is a full set of chromosomes containing all the inheritable traits of an organism. And as we observe our surroundings, we can see there are many different genomes in existence, creating very unique and incredible organisms. If it seems like your body is constantly working hard, it is. Living organisms continually produce new cells through a process called cell replication throughout their entire lives. And here's how it works. First, we start with our DNA double helix. Helicases are molecular motors that use the chemical energy of ATP to break hydrogen bonds between bases and unwind the DNA double helix into single strands. This process is used for both replication and repair of the strands. If unwound, a single DNA strand would be about 6 feet in length. And yet, it fits into a cell which can't even be seen with the human eye. Remarkably, these strands are kept from tangling as they are separated and duplicated. Next, these strands are copied by enzyme-based computers, creating an exact replica of each original strand. Over 30 different proteins are required for duplication and repackaging of cellular components during cell division. It's from this tiny, masterfully complex cell that life. Think about our own body having approximately 100 trillion cells, each group having a unique purpose, such as epidermis, the lungs, heart, skeletal structure, muscles, nervous system, digestive system, reproductive system, immune system, and the brain all working simultaneously to form this magnificent machine we call the human body. We begin to have an appreciation for the complexity, elegance, and beauty of life.